My name is Shri and this is Aditya and we are from Cunha Lab. And today we will be presenting our project, in vitro characterization of human epigenetic inhibitors, DNA methyltransfer inhibitors, and HDAC inhibitors on HCT-116 colorectal cancer cells. And by the way, in vitro just means it's happening outside the living organism in like a petri dish or a test tube. So this is a quick list of table of contents that we will be going through. Background. So in normal cases or cases without cancer, a methyl group, which is just a CH3, is added to DNA, and this is called DNA methylation. This methylation can alter gene expression and create transmissible or inheritable changes. In the case of cancer, methylation is associated in gene repression, meaning this represses tumor suppressing genes, therefore allowing tumor growth. In the case of DNMTs, these DNMTs are responsible for transferring the methyl group. DNMTs such as azacitidine or AZA are proven to be effective in treating myelodysplastic syndrome. Myelodysplastic syndrome is a type of leukemia or blood cancer. AZA improves the expression of the GADD45 gene, therefore leading to suppressed cancer growth. This is our research plan. We are researching the colorectal cancer, or CRC, which starts in the colon or rectum. It is the third most deadly cancer or third most common and second most deadly cancer. We work with epithelial human colorectal carcinoma 116 cell line, or HCT-116, and we chose decitabine as our control variable due to its potency. Through this use of decitabine, we plan on comparing to various DNMT and later HDAC inhibitors. The project's main goal is to compare various DNMT inhibitors that reactivate tumor suppressor genes by demethylating them. Methodology. We will be using MTT assays, qPCR, and Western Blots to continue our research. We will also be using cell maintenance to ensure that our HCT-116 cells are healthy and prepared for experimentation.
There you go.
Yeah, thank you. So the first step of our methodology is cell maintenance. So in order to study colorectal cancer, we use cells which are grown outside the human body and tested. And to grow these cells, we create media, which serves as a kind of food for the cells to grow in. And once the cells reach full capacity in their containers because they adhere to the flask, they must be given new space with new media, which is called cell splitting. And once we have too many flasks to maintain, we freeze down the cells and store them in the freezer for long-term storage, and so we can use them for future experiments. And when we want to use them again, we thaw the frozen vials and centrifuge them so that the cell pellet stays in the bottom, and then we can resuspend them in media and in a new container. And then we can grow them in cell culture again so that we can use them for immediate experiments. And now onto our cell viability assay, or our MTT assay, which is named after the compound used in this experiment. So the MTT assay measures cell metabolism, and this can be used to measure cell viability. And the first step of the MTT is to transfer the cells from their cell culture flask to the corresponding 96 well plate and adding the experimental drug to the cells as well. And once the cells are incubated, the MTT reagent can be added to the cells. And when the MTT solution is added to the cells, they react with the cell's mitochondrial enzymes to emit a color once it's dissolved in another substance like DMSO. And finally, we used a machine called a spectrophotometer to read the absorbency of each well, which is basically how we can tell this how strong the color is. And the stronger the color, we can assume that there's more cells that are metabolically active, and then we can compare this to our control groups, which are on the other wells, to determine cell viability. And the next experiment is RT-qPCR, which stands for reverse transcription qPCR. And this experiment measures the expression of a specific gene of interest that we want to know more about, and we want to know if it's being re-expressed or if the expression is lowered. And the first step of qPCR, like the MTT assay, is to seed and drug the cells. And the next, the mRNA is extracted from the cells using a kit. Since the qPCR works with DNA, the RNA must be converted into cDNA, or complementary DNA, which is why we use the enzyme reverse transcriptase to convert it to a complementary strand of DNA. And next, the specific primer, which is a section of the gene of interest, is added to the mix of cDNA, which binds to the proper areas. And then after that, we use a device called a thermocycler. The cDNA is replicated repeatedly by heating and cooling the strand through the steps of denaturing, annealing, and extending the strand, which is done by the enzyme DNA polymerase. And in this step, dyes are also added to the target sequences. So after multiple cycles of annealing and extending, the expression of the target sequence can be visualized. And this expression can be compared to a control sample and a housekeeping gene, which is a sequence of DNA that is present in almost all cells, regardless of the drug. And the final experiment are Western blots or immunoblots, which measure protein expression. So similar to the other two experiments, the first step is to drug the cells. And then after drugging, the protein is extracted by adding something called a lysis buffer, which extracts protein by breaking down the organelles and preserves the protein as well. And next, the protein is separated through something called gel electrophoresis, specifically something called an SCS page. So the gel serves as kind of a filter, which filters the protein based on its molecular weight, which is compared to a protein ladder, which we already know the molecular weights of. And as a part of gel electrophoresis, buffers are added to charge the protein negatively and positively, and allow it to negatively, and allow it to travel to the positive end of the gel, as well as to ensure movement of the protein to the other side. And next, the gel is transferred to a membrane, usually something called a PVDF membrane or a nitrocellulose membrane. And the gel and the membrane are kind of sandwiched together, and an electrical current is used to transfer the proteins from the gel to the membrane. Then the primary antibody is incubated with the solution and binds to the target protein. So this is an important step because the primary antibody attaches to the protein that we're trying to measure and see if it's being expressed. And then after the membrane is washed, something called a secondary antibody is added to bind to the primary antibody. So the secondary antibody allows us to actually visualize the target protein. So without this step, we can't actually see it in the imaging step. So finally, the membrane is visualized using imaging equipment, which can allow us to compare it to our control groups, like our housekeeping gene and the untreated cells. So for our results, we're currently working on qPCR and Western blotting, and we're also currently in the middle of our MTT assay. But regarding our qPCR, we're currently working on our mRNA optimization. So after we perform the RNA extraction after drugging our cells, we're supposed to check the purity of the RNA using a device called a nanodrop, which also records the absorbency of the RNA. And when we extract the RNA, we look at the graph to see if it's pure or not. So the goal is to achieve a value of 2 for these two numbers here, the 260-280 ratio and the 260-30 ratio. So the highest purity of RNA is required for the best possible qPCR results. So we're currently trying to reach these values. And so our future steps, we plan on doing flow cytometry, which tests whether the cells have proceeded past a certain point of the cell cycle, like the G2M checkpoint and interface, which would specifically check if the tumor suppressor gene is being reactivated by the drug. And next, we plan on doing fluorescent microscopy, which is used to analyze the apoptotic process in the cells to test efficacy of the drugs. So using this assay, we can also check if the drug is efficiently reactivating the genes. And finally, we plan to use a protein called CRISPR, or Cas9, which allows the gene to be reactivated without inhibiting the enzyme itself, but instead editing the gene sequence. But that's in the long term. And finally, we plan on switching from DNMT inhibitors to something called HDAC inhibitors or histone deacetylase inhibitors. So HDACs, they inhibit colorectal cancer in a different way by removing acetyl groups from lysine instead of actually inhibiting the enzyme like DNMT inhibitors do. And in cancerous states, HDACs reduce apoptosis and increase proliferation. And so HDAC inhibitors reverse these effects, which means they're effective in reversing cancer as well, treating cancer. So in conclusion, we plan to shift our research into new areas like HDAC inhibitors and the microbiome. And we plan to continue our research with these new assays like characterization and Western blotting. And the final scope of our project is to evolve into an FDA-approved drug in the future, following a pathway similar to that of 5-azacitidine and decitabine. And overall, we're hoping to expand what we know about these DNMT inhibitors and reach novel solutions to reverse DNA methylation in colorectal cancer cells. So we'd like to thank our advisor, Mr. Cunha, for being a wonderful advisor and inspiring our research and providing us with the resources to succeed. So thank you.
We would also like to thank Dr. Edward New for providing us with the drugs to experiment with, thus fueling our entire experimentation.
We'd also like to thank the lab technicians for ensuring our safety and well-being throughout our work while supplying us with the materials we required whenever we needed them. So thank you as well.
And lastly, but most definitely not least, we'd like to thank the Aspiring Scholars Directed Research Program, ASDRP, and the Olive Children Foundation for providing us with the resources and the opportunity to conduct this research.
Any
These are our
questions?
references in an easy QR code. Thank you. Do you have any questions?
So you said that DNA methylation causes basically causes the gene to the tumor suppressing gene to be suppressed. So what causes hypermethylation?
I'm actually not exactly sure. Aditya, would you like to answer that?
Yeah, there are like multiple reasons that can cause hypermethylation. Like most of the cancers that are detected, we don't know how they actually happen, but it could be due to epigenetic dysregulation or environmental factors, and also genetic predispositions. Like if your relatives have this type of cancer, or if they have a history of hypermethylation, that can also cause um, hypermethylation in patients.
Like specifically in breast cancer, a genetic aspect is mostly the reason.
Okay, all right, thank you.
There's a question in the chat if you want to take a look.
Oh, okay. Thank you.
You're welcome.
Aditya, I think that's for you.
Oh, what was the question?
Can you explain how you will use the fluorescence microscopy will test cell apoptosis?
Oh, I'm not specifically sure about the technique of fluorescent microscopy, but I know that it works with dyes, and those are added to the cells, and that can also be used to see which organelles are not working and stuff like that. But we have to do more research on that because that's a future experiment we plan on doing.
Are there any more questions? I don't think I see anything new in the chat. Oh. What are the long-term effects of decitabine treatment on HCT-116 cell viability and proliferation? Oh, okay, there's a couple. Okay, I'll answer the first question. So at this current moment, like, yes, the decitabine and AZA or azacitidine are both FDA approved, but they're not being used because they're very toxic. So yes, they do work in like demethylating, but it's not the best choice, which is why we're trying to test other ones. And then Aditya, how does this thermocycler work by denaturing DNA?
Does the okay. Yeah, the thermocycler has three different stages. It denatures the strand, and then it anneals the strand, which basically it adds more DNA, and then extension, it again it increases the temperature of the strand, and then DNA polymerase works to expand it. So in short, yeah, the thermocycler first it denatures the DNA. There's one more question. What will the use of flow cytometry on your project? What will the use of flow cytometry on your project? What does it test? Oh yeah, so flow cytometry, it can test cell viability, and also it can detect a DNA content and other stuff like organelles and specific stuff in the cell. So we can use that. So like the scope of that experiment is a lot, and that would definitely help our research. But we plan on doing that in the future. Yeah. And yeah, I see one more question. Are there any specific genes that decitabine affects that you're looking for? Yeah, we did some research and we found that there was a paper where this gene called MC353. I forgot the exact name, but it was proven that this gene was re-expressed after adding decitabine, and so we plan on also researching into that when we do our qPCR.
For the next one, so is everything needed to replicate DNA already in the solution put in the thermocycler at the beginning, or do you need to add things later? That's one of the things that, or one of the experiments that we still haven't done yet. So I'm not specifically sure, but I'm pretty sure you would have to add things later on, like to continue the process, because there are multiple stages to replicating. Does anyone else have any questions? Is that it? Okay, I think that's it. Thank you so much for listening to our presentation, and I hope you enjoyed it.